Good evening, or good morning, or good afternoon, depending on when you're watching this. Today we're going to talk about the light-independent reactions, and basically we're going to look at what goes in and what comes out and the entire process as a whole, so that you can get a better understanding of how it works and what the importance of it is. Now, before we get started, I'm going to start my little plug here, my Star Wars plug. So these are called the light-independent reactions. They used to be called the dark reactions. And so I have witnessed the full power of the dark side. Anyway, there's my Star Wars plug. And no, I'm not embarrassed. I love Star Wars. Okay, so yesterday, or in a previous lesson, assuming you watched it yesterday, we looked at the light reactions. And we determined that there's a series of reactions involving light, the photosystem 2 and photosystem 1, follow the bouncing electrons, and we get certain products, ATP, namely, and NADPH, which are going to be very important for the Calvin cycle. And the Calvin cycle is the light-dependent reactions, or the dark reactions, and that's what we're going to look at today. And the Calvin cycle is pretty important for life on planet Earth in general. Light reactions are too. The Calvin cycle is what takes the products of the light reactions and carbon dioxide and puts them all together to help make sugar. And sugar is the lifeblood of most organisms on planet Earth. At some point in a food chain, every organism is consuming plants. And of course, without those, animals, more namely us, wouldn't exist. Okay, so it's a pretty important process. We're going to look at this. We'll look at this in a little bit more detail. We're going to talk about some of the things that happen, and more importantly, we're going to look at the products that come out and the reactants that go in a little bit more detail. Let's get started. Okay, so the sugar generating process. It's very similar, although obviously there's differences, to the Krebs cycle in cellular respiration. Okay? What we start with, or what we essentially begin with in this process, is 3 molecules of carbon dioxide. Now, I know what you're thinking. 3 molecules of carbon dioxide isn't enough to make glucose, which has 6 carbons. Okay, so this is actually going to happen twice. Okay? So, essentially, Rubisco, which is an enzyme, is going to fix the carbon dioxide. Okay? And all that really means, for our intents and purposes, is that it's going to attach the carbons that are found in the carbon dioxide to each other. It's going to fix them. Okay? Now, this process requires quite a bit of energy. As you can see here, we've got 6 ATP here being turned into 6 ADP and 3 ATP over here being converted into 3 ADP. So for 3 carbon dioxides to be turned into 1 molecule of G3P and then to regenerate Rubisco, which has to happen for the cycle to be able to continue, we're actually going to use a total of 9 ATP. And then over here, we have NADPH, 6 of them in fact, being oxidized to 6 NADP+. Okay, so remember the light reactions, how we talked about we were forming ATP and NADPH? Well, to make 1 molecule of G3P, we need 9 ATP molecules and 6 NADPHs. Otherwise, the process doesn't work. And as I said before, 1 G3P isn't enough to make a glucose. Basically takes 2. So everything you see here can essentially be doubled. It's not quite as simple as that, but we're just going to keep going with that theory. Let's get into a discussion now a little bit more about specifically how you can break this process down. There are essentially 3 parts or phases to the Calvin cycle. And you can see them quite nicely labeled here. Phase 1 is carbon fixation. As I said, carbon fixation is where Rubisco helps take the carbons in the carbon dioxide and stick them together. Okay. Phase 2 is the reduction phase. This is where the NADPHs come in. As we know, the NADPHs, because they carry electrons, are good reducers. And NADPHs, because they carry electrons being good reducers, remember, reduction is a gaining of electrons. Grrr. Okay, so the molecules in this phase are being reduced because the NADPHs are donating electrons. At the end of phase 2, we have an output of 1 G3P. At the end of the reduction phase, we have an output from the cycle of 1 G3P. And that makes sense because we started with 3 CO2 molecules. Now, before the cycle can continue to make more G3P, Rubisco has to be regenerated. Okay, and that's where this phase is, regeneration of Rubisco. So Rubisco, in this part right here, is regenerated. You do not need to know the names of any of these molecules. It wouldn't hurt to know the name of Rubisco and G3P because that's a product. Obviously, you need to know that 3 carbon dioxides are going in, 6 NADHs are going in, and a total of 9 ATP are going in to make 1 molecule of G3P. And of course, when you use up the ATP, the 9 ATPs, they're going to turn into 9 ADPs, which are going to be recycled back to the light reactions. We're also going to create 6 NADPs, which again are going to be recycled back to the light reaction. Okay, so we've got 3. This process isn't that bad, actually. You've got 3 phases: carbon fixation. Rubisco fixes these 3 carbon molecules, sticks them together. Phase 2, reduction. Few chemical reactions that occur to turn these carbon dioxide molecules into G3P. Okay, and then remember, G3P is eventually going to be converted into some sort of organic molecule, OM, organic molecule. And then the regeneration of the carbon fixing molecule. Okay, so we have to recreate this molecule, otherwise the cycle can't continue. That's it. In a nutshell, that's how it happens. Okay? The most important thing to remember, though, is that the plant is trying to create organic molecules, most namely glucose or sugar. Okay, but it can make other things too. It can make amino acids, fatty acids, really whatever it needs. And that, in a nutshell, folks, is how the Calvin cycle works. Make sure you jot down some notes on this cycle, and please bring any questions you might have to class so I can help you out.